Good morning, all the delegates. Today, we will start our day 6, session 3 of Mr. Hanif Shaikh sir, vaccine researcher, international vaccine clinical, Seoul, Korea, on topic clinical development of vaccines in emergency situations. Now, first, I will introduce Mr. Hanif Shaikh sir. Regarding his educational qualification, sir is pursuing PhD Pharmacology Maternal Vaccine, Pune University, India. Sir completed his PG program in epidemiology from Indian Institute of Public Health, Delhi in 2018. Sir is also recipient of certification course on epidemiological evaluation of vaccines, London School of Hygiene and Tropical Medicine, London, UK in 2017. Sir also did his Masters of Business Management, Projects and Operations Management from University of Pune, India 2000 in 2014. Sir also a postgraduate diploma in clinical research management, IMSR India in 2013. Masters of Pharmacy, Pharmacology from Savitribai Phule Pune University in year 2006. Sir also have a qualification of project management professional and certification in monitoring and evaluation by IS Global Barcelona and WHO TDR. Regarding his work experience, currently, Sir is a researcher at International Vaccine Institute, Seoul, South Korea, since 2021. With his past experiences, Sir worked as an operations research coordinator at MSF Medicines Sans Frontiers, Doctors Without Borders India, Brussels cells from 2019 to 2021. He is also a high caliber investigator at CDSA THSTI India from 2017 to 2019. Sir also a WHO fellow and consultant and clinical development and regulatory at International Vaccine Institute, South Korea. Senior project manager at KEMHRC Pune. And sir is also worked as a research associate, medical affairs and clinical research at Ranbaxy Research Laboratory, Mumbai. Regarding his active contribution and expertise in the field, he is a dynamic and results driven professional with greater than 18 years of experience in epidemiological and clinical research, project management, and capacity building. He is having a comprehensive knowledge of vaccine development and implementation with proven expertise in leading project initiatives from conception to completion with a keen focus on aligning with international standards. He is also skilled in the end-to-end management of projects, including the project planning, monitoring, and that liaising effectively with local healthcare professionals. Adept at epidemiological data interpretation and dissemination with a strong track record in producing high quality reports. He has also demonstrated ability to foster collaboration with key international stakeholders, drive health policy advocacy, and secure funding to support mission critical projects. He is a proficient in supervising and mentoring junior researchers and interns, ensuring optimal team productivity and project. And Sir is having a variety of publications in national journals and international journals of repute. Regarding his awards and achievements, he is a recipient of registered member of Maharashtra State Pharmacy Council. He is a life member of Association of Pharmaceutical Teachers of India and American College of Clinical Pharmacology. Sir is a member of Global Health Network Oxford, UK, member of BACTI-VAC Network Birmingham, UK, member of Imprint Network London, UK, and member of American Society of Tropical Medicine and Hygiene since January 2023. So, with this brief introduction, I will request Hanif Sheikh sir to start session 3 on clinical development of vaccines in emergency situations. Please, sir.
Okay. So first of all, thank you very much for inviting me for this very wonderful faculty development program, and I'm really happy to accept this invitation when Mishal ma'am asked me if I can deliver some talk, because I was in academics for a couple of years, and I always like to go back and talk to my colleagues and share the new thing which is happening in industry. And it's really a great honor for me to come back and deliver a talk on very important topic of clinical development of vaccine, specifically when you are talking about the emergency situation. We had gone through very critical situation a couple of years back, and that was COVID-19 pandemic. Well, before I start my talk on the clinical development of vaccine focusing on emergency situation, I would like to give a bit of background why we are talking on this and what exactly we are doing to handle future pandemic situations. Uh, when there was a pandemic in 2019-20 onwards, we had faced a really critical situation. People were dying, and there was no vaccines or drugs available. In fact, we were quite fortunate enough to have certain platform developed, like a mRNA vaccine platform. However, getting that vaccine available for the population was far beyond our reach. Fortunately, within 100 days time, or I can say actually delivery time was quite more, within 1 year's time, we managed to deliver the vaccine to the population. But that delivery was very much restricted to certain area or developed countries like UK, US, Canada, and many other high-income countries. India was quite fortunate enough to have their own manufacturing facility like Serum Institute, Bharat Biotech, and so on. So we had not seen that particular shortage of the vaccine. But if you see the world situation, there is a high disparity of the distribution of the vaccine, specifically talking about the low and middle income countries. Again, African countries, there are certain areas where we didn't get the vaccine available or delivered to the population. There are people who are still waiting for the vaccine, COVID vaccine I'm talking about. It's been almost 3-4 years now. So to tackle such situation and to make a vaccine distributed across different part of the world equally, the WHO had developed certain program, and that program was mainly targeting to develop the vaccine within the country or within the region, like Africa or other Latin American or other developing countries. And with that, WHO and the other partners, they came up together and delivered the platform. And one of the activities they are doing is that creating a large manufacturing facilities in such countries. And Korean government was awarded as global training hub for the biomanufacturing. And I'm really happy that I'm the part of that global training hubs where we are identifying the potential manufacturers from these developing countries and training them so that they can develop their vaccine, not necessarily develop their vaccine, but they can produce their vaccine. If you see the data, they import 90% or more than that vaccine from the developed country, including India, but they don't have their capacity to develop their vaccine. Why I'm saying so? Because since I said the background that they are still waiting for the vaccine, and if such pandemic again happen in the future, probably they will be the most sufferer. And to avoid such situation, WHO and other important healthcare authorities to set up certain goals so that by 2014 they can develop their 60% of the vaccine within the region. And we are supporting them. And this slide which I'm using is part of that activity, that is a global training hub for manufacturing. We generally use this slide for the training of the researchers, scientists, manufacturers from such countries. Okay, so thank you very much to GDHB and IVI, where I'm working, International Vaccine Institute, which is a UNDP charter organization, non-profit, intergovernmental organization, and we are continuously supporting to developing countries to have their global health research data available, develop the vaccines, and supply the vaccine. But when we talk about the clinical vaccine development, we generally talk about uh, vaccination, because we believe that it's not a vaccine which saves a life, it's a vaccination which saves the life. Because developing a vaccine is definitely an important thing, but delivering to the people, right people, is again most important thing, right? So we are targeting to deliver the vaccine so that people will get the vaccine and they can save their lives. Well, these slides, as I said, are being used for the different training purposes. So we have certain objectives for such trainings. And the objective for this particular session is to apply knowledge to your own vaccine development program. That means the countries which are developing their vaccines. I'm using the exactly same slides, and 90% of slides will be more relevant to the faculty development program we are talking currently. And then use information gained today to create your clinical development plan in pre-licensure that will be acceptable to the regulatory authorities around the world. So we'll see how this regulatory affairs or regulatory approval process takes place to understand the requirement that must be considered when writing a clinical trial protocol. So how to design the clinical trials when you're talking about the vaccine development, and what are the other requirements. That is something which we'll see during this presentation. The content of this presentation is introduction, clinical development plan, regulatory requirements, adaptive trial design, because that is something which we generally talk about when we talk about the COVID-19 vaccine clinical trials or clinical development in the emergency situation. So adaptive clinical trial design are very important to match the timelines, and road to licensure and policy and conclusion. So introduction. I'm not sure whether I would be interact to the participant, but just in case, if you can see my slide, I would like you to either use this code or go to the website called menti.com and use code given on screen, and give me just one thought. When I talk about the COVID-19, what comes to your mind? And that I can share my screen. So through this in menti.com, I can interact with you. Okay, at least going to the first step, if you can follow this menti.com and use this code, if it's feasible for you, and there you will see 3 tabs requesting you to give 3 words. So you can type any word like infection or anything. So my question is that what comes to your mind when you think about the COVID-19? So I just wanted to understand the solution which you think in your mind when you talk about the COVID-19. So that makes things more relevant. Okay, I will wait for another 1 minute, and then I will share my screen, which will reflect the results and the thoughts which you had. You just need to either scan this code or just go to menti.com on your Google, use this code, and type 3 words which comes to your mind. I'll go to menti.com now. I'm not sure whether you can see my screen. I will put it on my screen. And I can see actually the words coming here. I'm not sure whether you're typing or not, but I don't see any response as yet. Okay, great. I can see a few responses here on screen. If you can also see those responses. So something like a vaccine, obviously that's what I was expecting when I talk about the pandemic situation. Pneumonia, for sure, this is one of the very important infectious diseases when we talk about the neonates or children. Register, yeah, that was a big register so as to collect the data. Infection, COVID-19, a vaccine sounds to be very prominent response so far. If you want to add a few more letters, please feel so. Okay. There are 6 responses registered so far. Great, but I think I got my answer. The vaccine is the most prominent answer, and that's why we are here to talk about the vaccine. Okay, well, I will close this tab and I'll go back to my slide. So, yeah. So when we'll talk about the vaccine development, we have to understand this entire journey. I know many of you might have uh, studied this about the vaccine development or drug development. It's not different than the drug development. But when you talk about the vaccine is quite expensive affair. It generally takes 10 to 15 years to develop a vaccine, right from the lab or the basic research to the clinical development or like getting a licensure. And the total expense, if you see, is more than 1 or 2 billions many times. And the success rate is quite low. When you start with the basic research, you have 100 candidates under screening, but when you actually deliver the vaccine or actually some vaccine which get the marketing authorization, the total number is just one. So that means that the success rate is very low, the risk is very high, and the time required to develop a vaccine is a really longer period. So we need to be very careful when we are talking about the vaccine development or we are dealing in the vaccine research. When you start with the vaccine clinical or vaccine research, we start with the exploratory research where we identify the basic antigen, which then studied in different settings like animal models or non-human primate models, where we identify its vaccine safety, efficacy, and further toxicity. And if candidate is found to be suitable for further stage of that is a clinical stage, we apply for the IND application, and then we conduct the clinical trials phase 1 and phase 2. That's what we are going to talk in this lecture. So the clinical phase of the development is very crucial because if you see the total budget, more than 60% of the budget is spent on the clinical development, and it's quite a lengthy process. It takes 5 to 6 years in total, and the success rate is again very low. Many candidates which gets approved in the phase 1 probably they could not succeed or sustain till phase 3. And again the important thing is that we are conducting a trial on the human participants, so obviously the risk is very high. So I will be coming back to this clinical phase of development after I talk about very important things which are some of the important definition, so that we will understand it better. I know this is not really very fancy part of this presentation, but we need to really go through it very quickly, so we will be on the same page, so we will not miss important aspect of the clinical trial. So when you talk about clinical trials, the clinical trials are the investigation on human subjects intended to discover or verify the clinical or pharmacological and or both pharmacodynamics effect on one or more investigational product, and to identify if there are any adverse effects. So these are basically trial conducted on a human participant, that is important, and safety and efficacy are the important things which we investigate. And there are different phases, as you know, that phase 1, phase 2, phase 3, and phase 4, which is essentially post-licensure phase. Good clinical practice is a set of guidelines, standard for design, conduct, performance monitoring, auditing, recording, analysis, and reporting, so that whatever the data which will be collecting will be standard and acceptable across different countries and different regulatory agencies. Investigational product is basically an active substance which we are considering for the testing in clinical trials, and it essentially include both investigational product and the reference material which we'll be using for the comparison. Regulatory agencies are the public authority or government agencies which actually monitors and provides certain regulation in which we conduct the clinical trials. So these are just standard definition. I wanted to just touch base before I move further. Okay, so coming back to the clinical development plan, vaccine clinical development plan, there are certain important steps which we have to consider, and those plans need to be developed very well in advance. When we start talking about the clinical development plan of any candidate vaccine, we generally develop a plan right from the phase 1 to phase 3, phase 4. We need to consider what is the target product profile. This is a very important document which anyone need to look into before developing clinical development plan. And what is it? So target product profile, that is TPP, is basically a desired profile or characteristic of the target product. If you're talking about a COVID vaccine, if you want to develop a COVID vaccine, so how that product will look like. So there are certain questions which we need to ask, and importantly, this document, that is target product profile, is mainly developed by the agencies like a WHO or similar agencies, international agencies, or many times also developed by the sponsors. But when WHO develops something, many times we have to align with those target product profiles, so that those, especially talking about the vaccine, so that WHO would accept our product, and when as and when required, they can help to distribute the product in different countries where the needs are. Okay, so there are certain questions which this target product profile addresses, and one of the question is, what will the final product look like? Like these three questions: what, who, and how. So one of the question is, what will the final product look like? In that, we have to talk about the indication, for what indication we are developing that vaccine. Similar like a COVID vaccine, we are developing for the prevention of the COVID infection. What is the mechanism of action of that, how that vaccine or that drug would work? What is the efficacy or effect size which we are targeting? Specifically, when we are targeting a COVID-19 vaccines, initially we thought that even though if vaccine could have 50% of efficacy, that would be quite enough to show its impact. And accordingly, the product was developed. Fortunately, many product had efficacy more than 60%, and that was well accepted by the WHO and other regulatory agencies. So we have to talk about the shelf life. If you remember, initially the shelf life of the vaccine was very less. Many vaccines were mRNA technology-based vaccine, so they were just having a shelf life 1 year or less. So when we are developing our vaccine, we have to say whether it should be 1 year, 2 year, or 3 years. So kind of those parameters should be set. Safety profile, whether there will be any interaction with the other vaccines or drugs, and drug formulation and dosing strategy, whether it's a single dose, multiple dose, or booster dose vaccine. That is something which you have to define. Similarly, for what target population we are preparing that product or vaccine, whether it's only for the specific gender, are we talking about only women or pregnant women, or we're talking about the children? Depending on the immune response or natural immunity, we can also decide whether this vaccine could be delivered only for the population below 5 years. You see that most of the childhood vaccine, or most of the vaccine in the program, are the childhood vaccine, because from the beginning only we have determined that this vaccine will be delivered to the children below 5 years. So that need to be also considered. Race, are we developing certain vaccines for the specific race, like Asian population or Caucasian population or any other population in the world? Because some countries have different responses, or some population have different responses. Then target countries, in similarly like high income countries, low income countries. Are we talking about specific climate zone? Because chikungunya you can find more in the Latin American countries. So accordingly, we had to say right in the beginning that this vaccine is being developed for the Latin American countries and similar regions. And another important parameter is, how will this product reach the market or the end user? And that what is our strategy for getting licensure, whether we are targeting WHO pre-qualification, or whether this vaccine will be delivered through the specific agencies. Like in many developing countries, the vaccine is not actually delivered by their routine mechanism. We are fortunate enough in India, we have our own mechanism, but some developing countries, the Gavi-like agencies, they support to the local government. They procure the vaccine directly from the manufacturer, help the government to deliver the vaccine to their respective areas, and then that's how they can vaccinate a maximum number of the participants. So similarly, who are the facilitators? Are right from the beginning, if we know that if we are targeting Gavi to procure our vaccine, accordingly we have to follow their set of rules and all the standards, right? So there are certain vaccines which are also delivered to the private market, and that need to be very much clearly specified in the beginning. Yeah, I will be talking about the WHO pre-qualification in my next slide. But yeah, so when we talk about the clinical development, as I mentioned, that we have to understand the TPP and then start actually developing our clinical development plan. And those plan should be very much clearly started in the beginning. We don't expect that plans won't change. A general rule is that no clinical study plan is complete until the last patient has been seen for the last time. So you always have to think of something new, and it can happen. The regulatory agencies may require more information during the trial, uh, and there could be some operational or conceptual things can arise which need a certain amendment to the protocol, and there are certain things which can ask to the sponsor to completely change their plan. So we should be not rigid when we're talking about the clinical development plan. Rather, we should be flexible enough, but we have to have our clear, defined objectives and endpoints. We should also have know right from the beginning what could be our potential sites, what are the total timelines through which we have to develop our conduct our clinical trials. Then what are the approximate resources which we require, and we should not really focus on a certain amount of the targeted countries or targeted agencies, but we should be flexible enough in terms of both timelines, cost, and other general ideas could be moved around those particular things. So in short, clinical development plan is very important, and we should have all those things assumed in the beginning. However, we should be flexible enough to adapt anything which may come during the clinical development. If you see the clinical development phase, it is quite long, 5 to 7 years, and many things can change during the 5 to 7 years period. And to be very honest, that is the most costly affair in entire history or life cycle of the vaccine development. So, as I said, more than 100 million US dollar is generally spent for the clinical development of single vaccine. Okay, so as you know that there are certain phases of the clinical trials, and those are really important. Similar like any drug trial, in vaccine also we have phase 1, phase 2, phase 3, and phase 4. But there are certain differences in vaccine, as we know that vaccine trial is being conducted on the healthy volunteers. So we have to be very careful, and there are certain ethical things which we need to consider throughout all these phases. So the objective of the phase 1 is to administer a vaccine candidate on humans, because this is the first in human trial. Before that, we had conducted trial on animal studies or different animals, and this is the first time we are testing the trial on human beings. So we have to be very cautious. There is a risk-benefit approach we have to see, and the primary objective is the safety and reactogenicity to assess the entire profile of the potential vaccine candidate. And then we are also checking secondary objective, the immune response, whether vaccine is producing certain immune response so that we can further continue the development of the vaccine. And if you see the study population, it's a very small comparatively to the other phase 2 or phase 3. We start with a very limited number of the participants, like 100, not even 100, or 10 participants, and those participants could be recruited in a stage wise. Like we start with the 10 participants initially with the minimum dose, and then we observe the safety profile. We wait for 7 days. We monitor the reactogenicity, the systemic reaction, the local reaction through dye record or through direct observation in our clinic, and then we collect those data. We present to the safety monitoring committee within an institute or within a sponsor, and then they study those data, and we may need to submit those data to DSMB, Data Safety Monitoring Board, and then they will give us approval to continue the further recruitment of the participants, and then we go on the remaining participants. So we have to do certain interim analysis before we go for the further recruitment, and that is a cautious process. So ultimately, the safety and well-being of the trial subject should always be the priority, and special consideration should be given to the characterizing risk and putting in place appropriate strategies to minimize the risk. So risk minimization is something which is very important in phase 1, though we are interested to understand the safety and immunogenicity when you're talking about the vaccine in phase 1 clinical trial. So I'll just give the example. I'm just also following my time, so I should not miss the timeline. I will just give one of the example of the VIDT typhoid conjugate vaccine, which we had developed in collaboration with the SK Biosciences, and we had conducted phase 1 study for the safety and immunogenicity, where less than 100 participants were put into the VIDT, our vaccine group, and we studied those participants for certain period, and then when we observe that the vaccine is producing immuno response as well as is found to be safe, we then continue for the phase 2 clinical trial. So I will just give one example of the one vaccine which we had developed thoroughly through different phases, phase 1, 2, 3, and 4. So that would help us to understand what exactly is happening when talking about a clinical development of any vaccine. Similarly, in a phase 2 clinical trial, we have to again take the same candidate mainly for assessing its safety. We also check its immunogenic response. However, here the other goals are to identify what could be the presentation, whether it's an oral vaccine, injectable vaccine, what could be the optimal dose, and what could be scheduled with a single dose, two dose, and what would be the duration between two doses, whether it should be 1 month or 6 month duration. So that kind of the data we collect in the phase 2 clinical trials. The numbers are comparatively higher. As a general rule, when you talk about a phase 1, we can say the 10 participants could be recruited, or in the multiple of 10 participants could be recruited. When you talk about phase 2, we can always say in the multiple of 100 participants. And when we talk about a phase 3 clinical trial, we always talk about the multiple of 1,000 participants. I'm talking about the vaccines. In drug, the numbers could be lesser, but vaccine, we have to have more number of participants. Why? I will also explain in my next slides. Okay. So the study population for the phase 2 is a few hundreds to 1,000, and we mainly recruit adult participants. Initially, when I was talking about the phase 1 trial, we also have to recruit adult participants, but we can also go to the other population like children and infant. But adult participants are mainly recruited in the phase 1. In phase 2, we have to again check the response in different population. So we can go for the age de-escalation, like starting from adolescent children and then infants, and then we see whether that candidate is safe enough to deliver to those particular population. Many times when we talk about the childhood vaccine, we are actually delivering that vaccine to the children. We are not intending to deliver that vaccine to the adult population, but despite of that, we have to study the response, immune response, or efficacy response on adults. So we have to start with adults in many times. Okay. So in phase 2, the study design is ideally a randomized control design, IP, that is investigation product, versus a placebo or active comparator. And we mainly collect the data for adverse event through the regular clinic visit or subject diary card, and we can continuously assess the immunogenic responses uh, across different studies. And if there are certain standards which are already established, externally validated, we are trying to recruit those standards in our phase 2 clinical trial. So the example from VIDD type of study, the phase 2 safety and immunogenicity study was conducted in Philippines, where 285 subjects were recruited of the age 6 to 20 years, and the trial was completed, and we then submitted our documents to the regulatory authorities for further approval of conducting the phase 3 clinical trials. Okay, coming to the phase 3 clinical trial, this is the most important phase of the clinical trial because it is the largest and most costly part of the clinical trial. Many times candidates get fail before phase 3 or after phase 3, and we generally believe that we have to assess the entire profile, safety, efficacy, immunogenic response of the candidate before phase 3. Otherwise, if candidates fails after phase 3, it will be a really big loss, right? So there are certain other techniques which we also follow, like a human challenge model, so as to avoid a failure later after phase 2 or phase 3 clinical trial. But that we are not going to talk about or in today's lecture. In general, phase 3, that's why I'm saying that phase 3 is very important, because the larger sample size, very costly clinical trial, and typically it is a multi-centric trial, multiple sites, multiple cities or multiple countries. And a golden rule is that when you want to establish efficacy of any vaccine candidate, we have to go for a randomized single or double blind clinical trial. And generally population is in thousands, like 10,000, more than 10,000. Sometimes it's 100,000 as well. So it is designed to confirm preliminary evidences, safety and immunogenicity accumulated during phase 2, whatever the responses we are collected in phase 2. And the goal is to assess the safety and efficacy of the product definitively. It has to demonstration of vaccine efficacy, which is fundamental for the licensure, because after only phase 3, whatever the efficacy which we can produce is going to be accepted by the regulatory agencies. And we also, during this phase, we also assess the adverse events or other safety concerns of the candidate vaccine, because this is a larger population. Sometime in phase 1 or phase 2, we might not have observed certain safety concerns because the population is very restricted. Some concerns, safety concerns which are very rare, could be observed in a phase 3 with a larger population size, and those need to be also produced to the regulatory agencies. So this is very important trial or important phase of the clinical trial. So as I mentioned, the gold standard for assessing the prevention of disease or infection in phase 3 trial is the prospective randomized double-blind controlled trial of a protective efficacy. This is something which we always do when we talk about conducting phase 3 clinical trial. We directly go and compare it with the existing vaccine. If existing vaccine is not available, we can compare with the placebo. However, there are certain other options, like when vaccines containing the same antigens are already in common use in particular area, and incidence of disease is very low in that region. The phase 3 clinical trials, although involving larger number of persons than previous phases, will be confined to the evaluation of the immune response and comparison with the recognized correlate of protections. As I mentioned in the first option, we are actually going and checking how much percentage of the reduction of the episodes of any particular diseases happen when we are comparing two different vaccines, either placebo or the reference vaccine. But in certain cases, we don't have that disease, the incidence of disease very high, so that we don't get the number of the cases. For example, if you're talking about maybe yellow fever cases in India might not be very high, but when you want to conduct in India, we will get out of 10,000 participants, will get maybe 10 cases or 15 cases, and that won't really help us to calculate the efficacy. So what we are going to do, we are actually going for assessing the immune response, and we had developed certain correlate of protection, and we will compare whether our vaccine can have that immune response compared to the other vaccines which are available in the market. There are another option, however, in sometimes there are no established and unequivocal immunological correlate of protection. It is important that some attempts should be made to estimate the effectiveness of the vaccine after its licensure and widespread induction. So correlate of protection is something which which is generally established after collecting a lot of data from the clinical trials. And in many cases, when the disease is new or vaccine is new, we don't have very clearly defined correlate of protection. So in such cases, we still conduct immunological studies, and after getting a licensure, we ask the sponsor to conduct the effectiveness study in the same setting, so that we will still collecting the some other effectiveness. Specifically, when we talk about effectiveness, we are also collecting the episodes of that diseases after vaccines is get introduced. Okay. Another and very important is non-infectives design. So when we compare certain molecules or some certain candidate vaccines with each other, not necessarily we have to go for the superiority design. We still check the immune response of our candidate vaccine and compare and say that whether that licensed vaccine, the immune response of the candidate vaccine is not less than some pre-specified ratio for when we compare with the licensed vaccine. So if it's in simple terms, if I can say that my vaccine is not equal to that available vaccine, but it's not less than that. It is within that range, within that ratio. So in that case, also this vaccine could be accepted. So there are different ways to conduct the clinical trial, especially in phase 3, to get the marketing authorization. And one of the very important is our head-to-head comparison to check the vaccine efficacy in the particular setting or particular population. Okay, but there are certain other options available. Okay, so we did conduct one immuno non-inferiority lot-to-lot consistency and safety study on Nepal population. If you see the number of the participants, it's just 1,800 subjects. Otherwise, if you had to conduct a phase 3 clinical trial, we have to use more than 10,000 participants. The number of the participants or sample size is basically defined or decided based on the actual incidence rate of the disease in that area. But fortunately, we had one vaccine available, that is type by TCB, and we had all the information related to the immunoresponse of type by TCB. So we conducted a non-inferiority lot-to-lot consistent trial, and based on that, that vaccine was authorized in that market. We did conduct another phase 3 trial in Philippines, where the immunoequivalence and safety was compared with similar other vaccines. Okay, so this was about phase 3. But there is one more study which we also conduct, that is called as first phase 2B study. It is more or less similar to phase 3 trial, but many times it is conducted after phase 2A and before phase 3. It's basically proof of concept before we go for the phase 2. And there is a similarity. Almost all the trial design and everything is similar, except the difference is in the lower bound of the 95% confidence interval for efficacy and power. When you talk about the phase 2B trial, it is always lower than, it could be lower than 0, versus phase 3, it should be above 25 to 30. And that's why we can compare that with this, whether it could be a phase 3 or phase 2B trial. When we talk about the lower bound of the confidence into 95% confidence interval, we have to always decide or discuss it with the local regulatory agency whether that is acceptable to them or not, or accordingly they set the lower bound confidence interval. In case of COVID vaccine, the regulatory agency decided that the lower bound confidence interval for 95 or 95% confidence interval should be above 30%, and the efficacy which they projected should be more than 50%. So when you refer to the targeted product profile, so the regulatory agencies at that time projected that when you are developing COVID vaccine, you should have this calculation. Your efficacy should be definitely above 50%. Otherwise, vaccine will not produce, or it will not produce enough impact. Okay, and the power is another area, another parameter. So in 2B, it is power could be 80%, but in phase 3 trial, it is always more than 90%, so that we will get a very solid analysis when you're talking about the efficacy or immunogenicity of our candidate. Okay, so this is another example of a dengue vaccine where similar clinical trials were conducted, and if you can see, if you can see the power and the lower bound 95% confidence interval for vaccine efficacy for phase 2B, which was conducted in Thailand, it was I'm not sure you can see. Yeah, it was less than 0%. But for phase 2B, which was phase 3, which was conducted in Asia and Latin America, it was above 25%, right? And the sample size is something very important. If you compare for phase 2B, the sample size was around 4,000, whereas in Asia it is 10,000, but in Latin America it is 20,000 and above, right? So this difference is mainly because of the incidence rate. As I said, lower the incidence rate, the higher the sample size, right? Here the incidence rate is 0.64 in Latin America, so the sample size becomes more than 20,000, whereas in Asia the incidence rate for the dengue is comparatively higher, so the sample size is lower. So that's where the sample size calculation happens. Well, moving further, phase 4 clinical trial, that is a post-licensure clinical trial, which is also known as post-marketing surveillance. This trial happens in actually real-world setting. Once we get a marketing authorization for our vaccines, we have to still continue collecting the data, whether it's a post-authorization safety data or post-authorization effectiveness data. So that data need to be continuously collected and monitored. And if there are any important signals which are collected, accordingly we can adjust our dosing or age, or we can relabel our product. So that is very important. Okay, So many times you might see that even after the product is licensure, after a few years, the regulatory agencies may ask us to withdraw the product from the market, and many times they can ban the product. So it is important to continuously collect the safety data even after getting a marketing authorization. The same thing applies to the drug as well as vaccine. Okay, what are the regulatory requirements when you talk about getting marketing authorization? We are developing vaccine mainly for two purposes. But first is in the interest of the public health, or second is obviously the companies want to make a profit. But there are certain rules and regulations. You cannot just make a profit as per your wish, and there are certain requirements which we need to follow when you are applying for the regulatory authorization or marketing authorization. There are certain guidelines which we need to follow, and some of the important guidelines are ICH, or we had in India we have uh, ICMR guidelines as well, but ICs are the international guidelines which we need to follow throughout the conduct of the clinical trials. So those are also considered as a part of regulatory. In India, ICGCP is also considered under the regulatory requirements, and there are different sections. I will not talk much about those guidelines because I believe that many of you might have studied these or know these about the different guidelines. Those are mainly categorized under quality guidelines, safety guidelines, efficacy guidelines, multidisciplinary directives, and there are different again sections under each category. And when you talk about the clinical trials, we always talk about the E6 guideline that is under E guidelines. Okay, and those there are certain principles. Those principles are based on the different codes or historical guidelines, whether it's a Belmont report or Declaration of Helsinki or other regulations which were adopted by different countries like the US and the European Union. But these ICGCP are very, very important uh, when we are conducting clinical trials in any country. There are certain revisions made. We are currently using R2 guidelines, but R3 has been already declared, which will be very much in use in next couple of months. Okay, principles of good clinical practice are clearly mentioning how these clinical trials should be conducted and how the safety, rights, well-being of the human participants should be protected, and how this manufacture investigational products should be manufactured, how the reporting, interpretation, and verification should be done, and whatever the process which we follow should be as per the approved protocol, which we have to prepare right in the advance. And the trial should be very well designed, and it should be executed as per the approved protocol. So when we talk about the clinical trial design, the trial should allow inclusion of the all populations despite making any bias. It should be scientifically sound enough to deliver the outcome of the trial, to deliver or to meet the objectives of the clinical trial. So the robustness of the scientific design is very important, and that is how there are certain agencies or stakeholders which also review the protocol and the trial designing. So trial objectives should be very well defined. It should be accurate and reliable. Statistical or clinical inference should be properly made in terms of in reference to the questions. We could have several objectives: primary objectives, secondary objectives, and exploratory objective. However, our primary objective is very, very important, where we are actually targeting whether it's safety or tolerability or immunogenicity of the product, and that should be very well clearly defined. However, we can have multiple exploratory objectives or multiple secondary objectives. Yeah. When you're talking about the designing of the clinical trial for the vaccines, target population and selecting or selection of the participant is key. Target population means if we how to conduct a trial in that particular area, we have to have certain eligibility criteria so that we will be very much clearly selecting the participant, and we always call it as control environment. So within that control environment, how do we maintain that control environment? So selection is one of the process. So we select based on some criteria like a gender, their pre-medical history, and then the age, and all those things should be very much defined. And additionally, we are also checking from which area the population is being selected and what are the incidence rates of those diseases for which we are developing vaccine. As I mentioned that like dengue is more prevalent in Asia compared to the South America. So accordingly, we have to be very much specific when talking about the target population. Target population ultimately for whom this vaccine is to be delivered, right? And up from that target population, we have to pick certain subjects. We cannot conduct the trial on all the population, but we have to be very much careful for when you are selecting certain subject, which will represent as a sample of that population. And that is a process where we have to include we have to consider the inclusion and exclusion criteria. Well, as I was talking about the inclusion exclusion criteria, there are certain examples how this inclusion and exclusion criteria should be defined, right, in terms of age and their willingness to participate in the trial, because the voluntary participation is important. If they say that we don't want to be part of the trial, and if you include them, that will be a clear violation of the ethical guidelines and your regulations. So we have to be very much careful. And the medical conditions. If you want to conduct the clinical trial of vaccine on certain population, like HIV positive or hepatitis population, where the participants are immunocompromised, so ultimately you will not get the same results where what you're expecting from the healthy population, right? And similarly for other medical condition could be considered. So yeah, so this is important. Selection of the control. Uh, I mean, we can conduct a clinical trial on a single arm group, but that is not how we will be coming with unbiased results. When we want to say that our candidate vaccine has a really good efficacy or safety, we have to always compare with a certain other control or the placebo group, and whether we have to select a placebo, that is a vaccine without any active ingredient, or the control or other reference candidate which is already available in the market. We had to take a decision whether we should go for the placebo or we should go for the reference. When we want to compare the efficacy, we are always interested in comparing with the placebo because it will give us clear differences between responses, right? But in many conditions, when there is a vaccine already available, we cannot conduct a clinical trial using the placebo. If you are coming to India and now saying that, okay, we want to conduct a clinical trial for a COVID vaccine using a placebo as our control, so there is a clear ethical alarm which will raised by the regulatory agencies and ethical bodies, saying that that means that another part where the placebo would be administered, or another group where part placebo would be administered, they will be deprived from getting the COVID vaccine, because this is the time where everybody can get the COVID vaccine, and generally we keep the participant in the clinical trial for 6 months to 1 year. So during that period, these people who are on placebo will not get the vaccines, so they are deprived from getting or information which is already available to public, right? So we should be very much careful, and though we get a very robust or solid result when we are comparing with the placebo, but we cannot, on ethical background, we cannot recruit those participants. Okay, but we always have to have a control group, either placebo or the reference. Then statistical consideration, what should be the power calculation, etc. Or are we going for some safety endpoints or efficacy endpoints, or are we interested in analyzing data at interim stage? So that is something which we have to consider while designing the clinical trial for the vaccine. Randomization is very, very important, because that is how we can convey that our design is not biased, and whatever the baseline characteristics which we are considering in our clinical trial will be equally distributed in both the groups, that is test and the control group. So it is a basically process through which we are equally distributing the baseline characteristics, and there will be clear comparison between both the group after the intervention duration or follow-up duration. Blinding is another thing. I'm just touching base to this point. I'm sure you might have heard this terminology, but these are very important. So as to avoid intentional or unintentional bias, we have to blind many times the participants, the investigators, or both of them, or sometimes we just need to have a certain group which is unbiased so that they can actually administer the vaccines, and such group are called as observer blinded trial. The remaining people who are actually collecting data are blinded, but the people who are actually administering the vaccine, because of the physical appearance of the vaccine, those people are not blinded. The remaining team is blinded. So we call as observer blind, otherwise a single blind, or when only investigators is knowing which treatment is being given, but the participants are not knowing. Double blind, when both investigator and the participants are not knowing which treatment was administered to the participant. And observer blind, where the entire team, including the investigator and the participants, are not knowing which drug has been administered, but there are certain groups which are administering the clinical trial drug, are knowing that what drug has been administered. So this is something which we follow. Then clinical data management is important. I will not talk much about that, but we have to take care that what of the data we are generating is of quality and integrity, so that there will be no further question asked about those important element. Otherwise, if we miss this part, we'll be actually destroying our study. Because whatever the study we are doing, at the end of the day, we are collecting the data, right? I mean, if I'm investing $100 million in my clinical development phase, so what actually I get at the end of the day is just few, not a GB even, what few MB data, right? And then I will be analyzing data and I will be generating a report, and that report itself is the outcome. So this data is very, very important, and that should be valid enough, and should have quality data. There are certain methodologies, techniques, certain obstacles should be avoided, and there are certain good data management practices should be followed to generate a good quality data, which is acceptable not only by the sponsor but across different regulatory agencies, because regulatory agencies also ask for the complete data set, right? Not necessarily the reports alone. Okay, so we'll just go to the next level, because we were talking about the adaptive clinical trial design. The presentation which I made so far is about conventional clinical development, where there are sequential phase 1, phase 2, phase 3, and after every phase, the regulatory agencies very cautiously review the data from phase 1, and then they decide whether you should be getting approval for the phase 2. And it takes a time. As I said, 5-7 years is a general time. And that is standard pathways. I'm not saying it's bad. We have developed many vaccines so far, and those were quite effective. But in fact, we had certain emergencies also in the past. COVID-19 was not the only emergency. In fact, there were so many other diseases which occurred in the past. However, we also had certain expedited or accelerated development system, and that system actually helped in COVID-19. Okay, but in general, the standard pathway is that we start with the research, preclinical research, then we submit that data to the regulatory agencies. They scrutinize that data and they give us they give us approval that okay, you can go for the phase 1. After phase 1, they ask the data. There is a lag in between. They then they give approval for phase 2. Then we identify new sites. It's a really lengthy process. And after phase 3, we get approval, and then we start manufacturing. Manufacturing takes another 2 to 3 years time, and then actually introduction of the vaccine, getting that vaccine introduced into the program, and actual vaccination to the participant is quite lengthy part, right? When we were talking about the emergency situation like a COVID-19, we actually wanted vaccine to be delivered as soon as possible. People were eagerly waiting when vaccine would be available, and for that we have to adjust certain timelines, certain things. Obviously those were risky. We had to take certain risks. When we're talking about the accelerated pathway, we had developed the antigen, the formulation, since we knew the basic genomic sequence of the COVID-19. I will talk more in detail, but we had a certain idea about the platform genomic sequence, so we conducted the preclinical study, and then simultaneously, rather than waiting for complete data from a preclinical study, we started a phase 1, phase 2, phase 3, overlapping on each other, and then by the time this actually phase 2, phase 3 trials were completed, the manufacturer took a risk, like Cinema Institute. They started manufacturing millions of the doses, and by the time phase 2 or phase 3 results were available, they were all ready to uh, deliver the vaccine to the government, right? And during that period of the manufacturing, actually administration was started. They also started conducting phase 3 clinical trials. So those all those important steps were overlapped. They were squeezed, condensed, so that we can match the timeline. Okay. So this is possible because of the adaptive clinical trials. We have been using adaptive clinical trials since many years, and though adaptive clinical trial is slightly different from the randomized control clinical trial, which we discuss in the conventional clinical development, but it is a subset of the adaptive RCD. The main characteristic is that one or more feature of the design can be changed during the trial scores based on the interim result from the data accumulated early on. I will talk about it. And they would allow researchers the flexibility to redesign trial procedure at interim stages. The flexibility can provide greater efficacy by reducing the number of patients required, allowing early decision to be taken to eliminate treatments that show little promise, and moving effective treatments to market more quickly. That is something a legal definition from the regulators. So it's a prospectively planned modification. In short, that adaptive clinical trial designs give us basically flexibility to make changes in the treatment. We can reduce or increase the sample size during the conduct of the clinical trial. That allows us to seamlessly integrate phase 2 or phase 3 together, and any modification in the clinical trial criteria or eligibility criteria could be possibly done during the phase of a clinical trial. In fact, in a conventional clinical trial or in conventional randomized clinical trial design, we have to set everything right in the beginning, and we cannot change in between. If we want to change anything in the protocol, sample size, we have to amend the protocol. We have to submit that to the regulatory agencies and ethics committee, get their approval, and then accordingly, it is quite a lengthy process. But adaptive clinical trial design, we have to set a certain criteria right in the beginning, so that if those things happen, we can modify our sample size, the inclusion exclusion criteria could be also modified, and the analysis could be possibly done in between. So interim analysis is something which is quite a regular process in adaptive clinical trial design, whereas in conventional clinical trial, we generally do it at a very less time. So when you compare the characteristic of the conventional clinical trial with adaptive trial design, conventional clinical trials are rigid, whereas adaptive are flexible. Treatments are in conventional, we could have 2 or 3 or sometimes more, but it is restricted always to the 2 or 3. Whereas adaptive, we can have multiple clinical trial arms. Protocol could be amended in conventional clinical trial, but in adaptive clinical trial, we have to have pre-specified changes. And if that happens, we can, without really going for the amendment, we can follow those changes. And then does little until trial phase is over and active throughout the trial. So whatever the independent role of independent data monitoring committee is quite frequent in adaptive clinical trials. So what I'm trying to tell is that adaptive clinical trial is something which allows us to squeeze our timeline to make a changes. If something is not working during the phase of the clinical trial, we can see how this initial data is giving us a signal, and accordingly we can modify the data. That means that if one treatment is not working well, we will reduce the number of participants in that treatment, and we'll put more number of participants in that arm where we are getting better results or more promising results. So that is something which we can adopt, and that will allow us to see or develop the profile of that candidate in a short period of time. Okay, I think I'm running late, so I'll just go to the next slide. So I'll just give an example of the clinical development of the COVID-19 vaccine, where we had this accelerated speed of vaccine development, where there was a urgency from the pandemic urgency from different stakeholders, and there was accelerated clinical testing needed, specifically how this vaccine is working, whether it's really producing sufficient immune response. You might have seen that there are different diagnostic test kits were also developed. Then there was a process developed by the regulatory agency. Specifically, regulatory agency is very stringent. We believe that they don't accept our requests, but they were quite obviously they were confident in what they were doing, but they were also flexible to adopt certain changes, and they made this expedited review process. Otherwise, initially, if you remember, in India, just to get a one clinical trial approval, we have to wait for 1 year. So now it is happening within 1 month, right? So there are certain funding mechanism available from the different sources, and then there are massive requests or massive demand from the population, the government, and with all these important factors, we developed uh, accelerated development of the COVID vaccines. Within 1 year of time, we could deliver this vaccine. So we talk about the COVID-19 vaccine had been actually developed in 100 days, though the vaccine was delivered a bit later. But within 100 days, we could actually develop the vaccine. It would have saved millions of lives and trillions of dollars. Achieving the 100-day goal will give the world's fighting chance of containing future outbreaks before they spread globally and reach pandemic proportions. So, based on this situation, actually, the SAPI had come up with a new strategy called 100 days. That means that if we get to know any pathogens, genomic sequence or complete profile, within 100 days we can actually vaccinate the first human being. Within 100 days we can develop the vaccine and get the regulatory approval, everything. So that is 100-day mission, and that is very promising. And nowadays we are working on new different emerging pathogens, so that will be pandemic ready. We can say the pandemic ready. So any pandemic happens in the future, we can have vaccine available if you know the genomic sequence of that vaccine based on the current regulatory pathways and mechanisms in terms of different platforms which are available, like mRNA vaccine platforms. Okay, I just wanted to give one small example where we had the different vaccines developed with the different timelines. If you see the example of the dengue, the yellow line, actually, we know about the microbial sequence of these dengue vaccine right in 1944, but actually vaccine was delivered quite late. In 2019, vaccine was delivered. No doubt we have vaccines for dengue now, but see the total time period required to deliver the vaccine. And in case of the COVID, this highlighted part is we got to know about the vaccine genomic sequence late 2019 or early 22, and by the end of the 21, we were, or beginning of 2021, we were already with vaccines available for the delivery. So within a very short period of time, we could actually deliver the vaccine. And this is how the things work, that we have to adopt these adaptive clinical trial design, and that needs a cooperation from the different stakeholders, including regulatory agencies, funding, and all, right? Okay. Yeah, I will also, I mean, this is more or less same how this vaccine is being developed, and how the preclinical stage, after preclinical stage, we can actually um, overlap the different clinical trial phases, and within 10 months of time, we have delivered the vaccine. Okay. Okay, so once we have conducted clinical trials and the vaccine shows a good or promising result in terms of safety and efficacy, we have to get the licensure for the vaccine, and policy need to be formulated so that the vaccine could be delivered to the right population. And there are certain things which need to be prepared, right from the robust and comprehensive CDP with the high quality data. That gap analysis at the end of the phase 3 clinical trials need to be done, and then the risk management plan need to be prepared, so that we will be still continuing the data for the efficacy and safety after marketing authorization, and submit the full CTD in the right format as per the regulatory agencies which we are targeting, like USFDA, EMA, etc. So we have to submit these dossiers to the regulatory agency so that we can prepare the vaccines, or we can get the marketing authorization. And these are the different models or data which we require to get these marketing authorization. We have to submit this data in the typical format so that we can ask for the vaccine. But yeah, apart from regulatory authorization, we have to also get some support for the vaccine introduction into the population or the program. And there are different agencies, mainly the WHO agencies, which gives you support right from the identifying the target product profile, which we discussed in the beginning of this presentation, and till the time we deliver the vaccine, we have to get the data reviewed. What are the information which is coming up would be reviewed by the agencies like WHO. There are different bodies like a SAGE, piggyback, etc., and then they help us to, or they guide us to complete our documentation, and accordingly they help us to create a policy so that local governments or international agencies can adopt those data, and that vaccine could be easily delivered to the population, the targeted population, right? Okay, so pre-qualification is one of the very important thing where we want to deliver the vaccine to the certain countries, developing countries, which could be easily uh, adopted by the agencies like Gavi and other UN agencies. So we have to get the vaccine WHO pre-qualified, so that maybe in some cases, if that vaccine doesn't have the local marketing authorization, but in emergency situation, that vaccine could be immediately adopted and delivered to those population. So these are the important things which they ask. They want definitely quality of the data, both in terms of manufacturing and the clinical trial. They do inspect those sites and then confirm that the trials or the manufacturing was conducted in a proper way, and then they give a WHO pre-qualification. Well, so in short, we can say that before we start making any clinical development plan, we should check the TPP, that is target product profile, and all those plans should support that TPP. Identify your base case at the function and non-functional correlate of immunity. We can definitely collect the data about the correlate of protection, and then we can decide whether we should go for an efficacy trial, and what are the correlate of protection are we going to compare with the other candidates' immune response. Then phase 1 should be small with sentinel subjects, staggered enrollment, and safety lab assess would be done. Then phase 3 should select dose, regimen, provide safety and immunological data, have immunological go no go criteria for entering into phase 2b or phase 3 that are related to the correlate of protection, because anytime when you are developing vaccine, we have to set certain go no go criteria. If that happens, we have to stop the trial, like stop trial stopping criteria. We generally call it go no go criteria. Then pivotal efficacy studies, that is phase 3, are dependent on good estimates of incidence of disease, or must take into account the uncertainties which may happen anytime. Importance of interaction with multiple stakeholders, including NRAs, WHO, PTO, policymakers are important. That's why I showed in the last slide. These are the different stakeholders which are actually helping you, guiding you to collect the data or design your trial so that those data will be acceptable, and ultimately the vaccine will get a marketing authorization. And importantly, accelerated clinical development plan in COVID-19 era, vaccine developed and licensed in less than 12 months for European Union authorization, but post-authorization studies, that is example, booster, mixed match. If you still remember, we are also asking for the booster study, and then you might have noticed that okay, so we can notice that there are certain vaccines which were delivered, like one vaccine was given from the mRNA vaccine, second was from the Bharat Biotech vaccine, right? Initially, you might have received a Covishield, and then Bharat Biotech mix and match. You might have received the totally different booster dose. So such studies were actually conducted before we recommend such mix and match or booster, or we may come up with another 2 years booster dose, something like that. So that we are still continuing the long-term follow-up of the vaccine and seeing that how long that protection can sustain in those population who had received the vaccine in the initial stage. So that is something which was conducted in accelerated clinical development, and 100-day mission set by CP to prepare for the next pandemic is something which we would consider as our regular strategy to get ready for the future pandemics. So this is how we have delivered our vaccines for the during COVID time. And in this lecture, I just to go back and check what we discussed. I think we did talk about the vaccine development program, information gains today to create your clinical development plan. I mean, obviously, I'm not sure how many of you are seriously considering developing clinically or getting into the clinical development of a vaccine, but still, it is very important when we talk about the development of any vaccine or the drugs, and how these regulatory authorities consider different data. So we have to have that knowledge, and I believe we did have certain knowledge about that. and understand the requirements that must be considered when writing a clinical trial protocol. When you talk about the clinical trial protocol, we'll mainly discuss about the clinical trial designing, what are the different inclusion exclusion criteria, what are the comparators we should consider, and objectives and outcomes. So I hope it was delivered to the level of your satisfaction. And if there are any questions, I will be happy. And I think I completed right in the 60 minutes. Thank you very much.
Yes. Thank you, sir, for your session. Sir, some delegates have questions.
Yes, please.
Yes. Question from Dr. Vijay Lamole. How much time it takes to develop vaccine in emergency situations?
Well, so that depends on the vaccines basic information. If we have clear data about the vaccines microbial sequence and genomics, especially genomic sequence, as per our 100 days mission, we are all targeting to develop a vaccine within 100 days. But in general, we take at least 5 to 6 years to develop a vaccines. I had given an example of COVID-19 where we had developed vaccine within 1 year, or less than 100 days actual development of vaccine. But if such situation arise in the future, I think, if we know the genomic sequence of the vaccine, especially any not a vaccine, the pathogen, we can develop a vaccine within 100 days. That is what we are targeting. Okay, but practically getting all the population that vaccine would be challenging because introduction is a different challenge. That's what I mentioned, that in some regions like Africa where they are still waiting for the COVID vaccine. Probably in India we may not have that. We may not have to wait that long. But yeah, to be very specific in your answer, we can develop in a very short period of time in current situation. 100 days is that's what we are targeting as per the SAFE mission.
Thank you. Next question from Durgesh Kai. What is the current status of malaria vaccine across the globe?
We have developed a malaria vaccine. It has been launched in some sub-Saharan countries like Burkina Faso, Ghana. Serum Institute of India has developed one vaccine as well. They are also manufacturing vaccine, in fact. So we have vaccines available for malaria. Okay, that may be available. I mean, maybe available in India, but in global situation, we have a vaccine developed.
Next question from Zaki Iqbal. How clinical trials is carried during development of vaccine in emergency situations?
Okay, so clinical trials, as I said, that's a part of the clinical development. So, emergency situation what we face in the clinic, COVID-19, we conducted a phase one, phase two, phase three in a very compressive or condensed manner. We conducted phase one on 100 participants, and rather than waiting for the results from the 100 participants, we initially analyzed the data from the 10, 15 participants, and immediately we analyze those data, submitted to data to safety monitoring committee or DSMB, and after receiving approval from them, we immediately started with phase two. Similarly, rather than waiting the complete data from the phase two, we did an interim analysis in phase two, and based on interim analysis, we started at phase three. So these clinical trials are conducted in a very condensed manner, so as to save our timing. And adaptive clinical trial design was very much useful so that it helped us with the group or the candidate vaccine is really having desired efficacy. If you're talking about how clinical trial operation was done or how those clinical trials were implemented, I have answered for that. That means that during pandemic situation when there were actually no access to the population, we did have certain decentralized clinical trials where we recruited the participants, but we rather than inviting those participants to the clinical trial site, we did collect data very remotely. The data was actually monitored by the sponsor and different regulatory agencies remotely. So during those restriction or during the moment restriction, we did manage to complete our job responsibility rather than waiting for the all freedom for the operations. I hope I answered your question.
Sir, next question is from Rashmi Misal. Are all the stages of vaccine development strictly adhered to during development of vaccines in pandemic situation?
Yes, it is important because we are not skipping any stages, but rather we are overlapping the stages. We have to have to follow certain stages, but we have modified those in a such a way so that okay, there are certain things which allows us to integrate the phase one slash phase two slash phase three, so that rather than conducting those clinical trial in totally separate stages, we can possibly conduct in a one seamless phase like phase two slash phase three, and whatever the data which we collected during initial phase is immediately used for conducting phase 3 data in a same protocol. So that is also allowed, but we are definitely following all the stages, but those could be integrated, seamlessly integrated.
So next question, how to ascertain safety of vaccine developed during emergency by accelerated development approach? How justifiable is this approach as COVID vaccine was linked with sudden cardiac deaths?
This is a very important question, and that's why we are still following these COVID-19 participants. There are certain cases which are also coming up. No doubt, we are taking certain risks because letting people die of COVID-19 is more risky than having a vaccine which has certain well established safety limits or established safety profile is much better approach than that. But yeah, I understand this is ethically acceptable, and vaccine during the shortened phase we did have certain data, but we still believe that those data could be further, those participants could be further followed up for collecting more safety information. And we are also, having few signals coming up. Those signals are not very common, very rare signals, but still that is important. I mean, when Modi government actually introduced a co-vaccine in 2021, that was actually introduced in a clinical trial like phase. So they were still collecting the safety data when the people were actually vaccinated. Hundred thousands of people were actually vaccinated. We collected the data. There were certain debates on whether it's ethical or not, but it was a policy decision taken after considering different important bodies, including WHO or even local bodies, PDVAC and all. And it was I can say, a conscious decision taken to vaccinate the population rather than people dying because of the disease. So there are certain safety concerns, but I think that's why we are still collecting data. But those are very minimum. Those are very rare. Okay.
Thank you, sir, for your valuable insights on clinical development of vaccines in emergency situations. Sir, thank you so much. Now I request you to please accept our certificate as a token of gratitude on behalf of our college, MSG Copper. Please, sir. Thank you once again. Thank you so much,
Thank
sir.
you. Thank you very much. I really appreciate for your patience listening and the questions. Those are
Dear
really important.
delegates, yeah, thank you. Dear delegates, we are going to online for valedictory session immediately. So please you join kindly. We will share the link of valedictory session immediately.